Thank you all for joining us today. As you can see the title, we're going to have a presentation about the facts of COVID-19. Every day is always changing. Monday we hear one thing, Tuesday is something else. So we're just going to get all of those facts together. And we have our wonderful, wonderful Dr. Schlam to do this presentation for us. So if you just wait a little while longer while we get things together, we really appreciate that. And we're going to get started. Thank you all for coming.
My name's Everett Schlam. We're going to be talking about COVID-19 vaccines today. I have nothing to disclose. My college roommate actually is the head of the virology lab at NIH and the Vaccine Research Center at the NIH. Coronavirus has got its name because of the corona surrounding it. And they cause about 20 to 30% of the common cold or coronaviruses. Unfortunately, COVID-19, a coronavirus, can cause in a significant number of people systemic damage, not just to the lungs, but also intestines, brains, heart, blood vessels, and kidneys. And we've identified a post-acute sequelae of SARS-CoV-2, even in people with mild or asymptomatic symptoms may develop prolonged symptoms after COVID-19 infection. We could see that death rates and complications are much higher in the elderly and also in certain populations such as Hispanic and Black populations. So there are several what we call vaccine platforms. We're going to focus on the ones that are clinically available today, which are the mRNA and the DNA vaccines. In the vaccine development, there was no steps that were not included. So it was done the way we always do it, preclinical phase 1, phase 2, phase 3, and then the post-clinical phase 4. The reason that we were able to get the vaccine out so early is they overlapped these phases because the government supported that. Usually the phases are not overlapped because there's a lot of financial risk to proceeding on. So that's how these were developed so quickly. So the mRNA vaccines, that's the Pfizer and Moderna vaccine, basically the genetic code, the mRNA that tells our bodies how to make the spike protein. It's isolated, it's engineered, it's put in a lipid nanoparticle, and then it's injected into the arm of the patient. The mRNA gets into your cells, does not go into the nucleus, but it goes into the ribosomes and it tells your cells to make the spike protein. The spike protein goes to the surface of the cell and that elicits an immune response. So a very novel way for the vaccine and highly efficacious. Non-replicating viral vector vaccines, those are the vaccines like AstraZeneca and Johnson & Johnson in our country. They take a virus and they pretty much make it so it can't infect a human. Then they put the DNA code into that virus. This is so in the case of these vaccines, they use the chimpanzee virus in the AstraZeneca, and in the Johnson & Johnson, they used another type of virus. And what they did is they put the DNA code in, they inject it into your arm, and then this gets into your cell, and the DNA codes for the mRNA that codes to make the spike protein. So you end up making the spike protein. It's just an extra step. So the DNA vaccines have an extra step because that the DNA makes the mRNA that tells your ribosomes to make the spike protein. This vaccine is not available at the Novavax, which is this is done more traditionally. So they actually manufacture the spike protein outside the body, and then it's injected with an adjuvant, something that boosts your immune response to the vaccine, and then you get an immune response. So this is called a protein subunit vaccine. We have HPV vaccines, a subunit protein vaccine. We have several vaccines that use this technology. Novavax was supposed to come out soon, but it's been delayed because we have so much of the other vaccines available. The non-replicating viral vector is similar to what I had mentioned about the other DNA vaccines. So efficacy and safety was shown in multiple trials for all 4 of these vaccines, and we'll get into a little more detail with that. So you can see that these are very large trials for the mRNA, Pfizer, and Moderna vaccines, with over 40,000 people in the Pfizer trial and over 30,000 in the Moderna. These are the phase 3 trials, and they had a pretty good amount of different types of ages and ethnic backgrounds.
Not really.
It's hard to enroll people in these trials, so they try to mimic what's in the general population. So in our area, this may not be, but this was done over in different, many different areas. So for a vaccine trial, it's pretty good. You may want it to see more, but it's enough with this number of people, and even 13% is enough to prove clinical efficacy because of the large number of the trial. Because sometimes it's difficult to enroll different groups. That's the challenges with these trials. They went out specifically to try to enroll many different backgrounds. It's challenging, especially to do it in a quick period of time. So the efficacy of this vaccine was outstanding. This was the initial trial. Both were about 95% effective in preventing illness. Not quite as effective for AstraZeneca and Johnson & Johnson. And this is more of the breakdown of the Pfizer. And we'll go over some of the safety issues. But by far the most common is pain, redness, and swelling at the injection site. And a substantial number of people also get headache, low-grade fever, achiness, which is self-limited, usually resolving in 3 days. And the adverse effects with the Moderna vaccine were also similar, with a very high efficacy. This is the Johnson & Johnson, the human adenoviral vector vaccine, and also a study over 40,000. And you can see the efficacy not quite as high, but still very high for severe disease. And this is the AstraZeneca. We're not using AstraZeneca in our country, but it's being used in other countries. Efficacy not as high, but still very good for severe disease. And this is the vaccine, the Novavax, which is the subunit vaccine, which is uses similar technology to some of the other vaccines that are available. And again, you could see the efficacy was very high. We hope to see this available in our country in the fall. So in these trials, I went through that quickly because I want to have time for questions. The efficacy was far better than the expectations. 95%, nobody expected the vaccines to work that well, even the most optimistic scientists. So that was a real slam dunk for these vaccines. And this just shows some of the data of how effective some of the other vaccines in other countries are that we're not using in our country. And this is important. So really, global immunization is really, really important with this, and the cost gets to be an issue. As you can see, some of the vaccines are far less expensive. And this is a slide just showing which countries have the highest rates of vaccination. This is data from April. So the safety data analysis is really very important. And in these large trials, there were no signals of severe adverse events. Of course, the phase 4, where we have many millions of people vaccinated, sometimes things will come up, and we'll take a look at that. And this is just showing the Pfizer and Moderna vaccines, that it's not uncommon to have, very common to have injection site pain, redness, and swelling, and also to have headache, muscle aches, joint pain, fever, and chills. Can happen after the first or the second shot. And this is a slide just showing the comparison to flu vaccine and Shingrix. And you can see that the side effects are slightly more with the mRNA vaccines, but some of the side effects are similar. So the safety is, of course, a top priority with these vaccines. And these vaccines are really monitored very carefully in very unique ways. So that we're going to go over each of these: the vaccine adverse event reporting system, the vaccine safety data link, CISA, and BEST, and VSafe. So the vaccine adverse event reporting system, that's a passive reporting system where anybody can report. The doctor, the patient can report any type of problems. And then we're looking for any signals. The vaccine safety data link is actually more powerful. That's a collaborative effort, the CDC and some very large managed care organizations, close to 10 million people. These people are followed prospectively. So they're following people who got the vaccine, people who didn't get the vaccine, and they're looking for any signal for adverse events. And then the vSafe, some of you may have done this after you got the vaccine. You are sent a text message to see how you're feeling, and the CDC is monitoring patients who got the vaccine to see if they're having any adverse events. That's a novel approach. It was just started with this vaccine. CISA is really unique. If you have any questions about COVID-19 or the vaccine, you could email them or call them, and they'll get back to you usually within a couple of days. I think it's a very useful email. I've emailed them several times. So Johnson & Johnson had a signal towards vaccine-induced thrombocytopenia. This is a rare side effect, almost always in women between the ages of 18 and 49. AstraZeneca, which is the other viral vector vaccine, a DNA vaccine, also had that noticed vaccine-induced thrombocytopenia. And so this seemed to be higher than what would occur in the population normally. So it was determined that this CVST, cerebral venous sinus thrombosis, is very rare, but it can be clinically serious. And the rate was so low that the benefits of the vaccine far outweigh the rare risk of this occurrence. So another thing that was noted with the Johnson & Johnson vaccine was the possible association with Guillain-Barré syndrome. This was from the VAERS database, that there were 100 cases of GBS after seeing the J&J vaccine. That was after 13 million doses of the vaccine. 95 of the 100 people were hospitalized, one died. So the background rate of GBS per year in our country is between 3,000 and 6,000. So it was not sufficient to establish a link, but it was worrisome, meaning that approximately 1 in 100,000 may get GBS. So even though you can't establish a link, there might be some association because it's higher than the background rate. And anaphylaxis with the mRNA vaccines, again, the rate very low, probably around 2.8 to 2.5 per million doses with Moderna, and maybe about 5 per million doses with the Pfizer vaccine. And that's why we observe people after getting the vaccine for this very rare side effect. And there are some recommendations of people who've had a severe reaction with anaphylaxis after one vaccine. You wouldn't give another, but the rest are just precautions to vaccination. If you have people with severe allergies or problems with other vaccines, you would want to observe them for at least 30 minutes after the vaccine. Why this rare effect of anaphylaxis occurs is unknown. So they've been trying to identify why that might happen, but it's an extremely rare side effect. So this is just the Israeli study, and we have a lot of the data from Israel because they got a large percentage of their population vaccinated, and they were very careful following them. And it showed a very effective preventing asymptomatic disease. This was before the Delta variant. So it's tough to make predictions, especially about the future, and especially with COVID-19, because as Ashley was saying, things seem to change on a daily basis as new data comes in. How COVID-19 variants happen. So interestingly, the coronaviruses don't mutate as much as like a flu virus or other viruses. But when you have a huge number of people being infected, these viruses make about 1 billion copies in a human. And so you're going to have some mutations. And even though rare, some of these mutations may become more infectious, more harmful, to humans. And so that's what we're worried about. That's why the push to get people vaccinated as quickly as possible. That'll also protect people and cut down on the number of variants. So these variants, most of them are not problematic, but as we now know with the Delta virus, this one is more infectious and perhaps more harmful to humans. They label these in a different way. So the lineage now is alpha, beta, gamma, delta, lambda. So that's just naming the variants as they occur. So the recent data with Delta is it's far more infectious than the Alpha strain, which was the strain that took over in England and took over in our country. So as opposed to the previous strain that affected about 2 to 3 people after one person was infected, the Delta strains can affect up to 8 or 9. So it's much, much more infectious. So the good news is that our vaccines are very effective, about 90% in preventing severe disease from the Delta, but perhaps not as effective in preventing mild disease or viral load. What about a booster dose? Well, we're waiting for the CDC and the FDA to make decisions about those. CDC has supported a 3rd dose of COVID-19 vaccine for immunocompromised patients, but it's not yet FDA approved. Pfizer has some data showing waning immunity of its vaccine after 6 months, with a 5 to 10-fold increase in antibody titers following a 3rd dose. And the increase in antibody titers is actually higher in the elderly than the young. Israel and England are endorsing a booster vaccine in certain populations. And so the FDA and CDC are evaluating the data, and they'll be deciding whether booster dose is necessary probably before the fall. At this point in time, it's essential to get unvaccinated people to receive 2 doses of the COVID-19 vaccine. The Delta variant, 1 dose is not nearly as effective as 2, so it's essential that they get 2 doses. So, as I said earlier, there's a lot of uncertainty as far as will there be new variants, will we be getting a booster, and so forth. One thing we don't have is a correlative protection. So you may be familiar with a hepatitis B titer or a measles and mumps rubella titer, saying if your titer is a certain amount, then you're protected. We don't have a correlative protection with COVID-19 vaccines, but I know that the CDC has been working on this furiously, and hopefully we'll get that. There's no guarantee. There are certain infections that we're not able to get a correlative protection. So is it possible, can we get a universal coronavirus vaccine? So this would be ideal because this would protect against variants or anything. So there are proteins on the coronavirus that are stable over time. And so there's around 4 or 5 different vaccine platforms being investigated now. And I think one clinical trial is already in phase 1 and phase 2, trying to develop a universal corona vaccine, which would be ideal. We've been trying to get a universal flu vaccine for many years also, and we don't have that yet. But there's definitely work on this, and we'll hope to see that, but that probably will not be available for at least 3 to 5 years. So you can see there's, even since 1995, when these residents graduated from our program, we've had several epidemics, not pandemics like COVID-19, but we've had several viral issues. 2 of the previous have been coronaviruses, right? And you could see that the number of vaccines that we give, this is the childhood vaccine shows this has increased enormously since 1986 to 2019. The interesting thing is there's actually less antigens in all these other vaccines than there was in the few vaccines that we gave in 1986. And the reason is they'd be able to purify the vaccines, and so they're less reactogenic. So unfortunately, pandemics are not unusual in history, and we've had quite a few, and some of them have been incredibly devastating to the world population. Our most recent one was the Spanish flu in the early 1900s, which killed 500,000 of our people, and is estimated to kill between 30 and 50 million worldwide. So we're going to be continuing to see a lot of data and a lot of information come in. This is just one point in time, but we have to be alert to new information and keep up with everything. And most importantly, encourage our patients and to get vaccinated. So I went through that quickly because I wanted to leave a lot of time for questions and any things that you had concerns about.
I have a question.
Yes.
Are you aware of any studies being done specifically for immunocompromised individuals with primary immune deficiencies?
There are studies being done. I don't have the actual data on them on many different immunocompromised populations. I don't know specifically about which one you asked,
Okay.
but there are a lot of studies being done. As you pointed out, the immunocompromised population is very, very heterogeneous. So it's not, there are people who have different types of immunocompromise. And so in Israel and in England, they've already started to immunize immunocompromised patients because they're at 10 times the risk of complications if they get COVID-19.
Yeah, I am immunocompromised, which is why I'm still working from home and will be throughout the end of the year. We have cold and flu season coming. We have the variants, so on and so forth, and it's been troubling to not really be sure whether the vaccine. I had the Moderna,
Good.
earlier this year, whether it's really protecting me or not. I had the spike protein antibody test, and it was really kind of inconclusive.
Did they give you a number on the spike protein?
No, you know what? I didn't get a number. My immunologist and I, we went over the results. I'm reading it. I'm looking at, the report, which of course is like a different language to me. And basically his response was, you've had somewhat of a response, but not one that he's comfortable with. So I am pretty much, my behaviors and actions are pretty much as if we're still in the height of the pandemic,
Yeah.
So I was just curious.
So there's so we have two types of responses, right? We have the cell mediated and the humoral. So we measure antibody levels, which are just one type of response. So that's the B cell response. So when we get exposed to the virus, after you got the vaccine, you get a memory B cell, and then you have these antibodies floating around. And what happens is the antibodies can latch on to the virus and prevent it from getting into your body. But there's also a very robust, what we call T cell response that we don't measure very often because it's much more expensive than measuring antibodies. Right. Those are T killer cells. So what they do is they attack the cells that are infected with the virus and they prevent severe illness. So we do think that there's a very reasonable and good T cell response still in people. The FDA and the CDC are waiting for more data. This is not evidence-based what I'm about to tell you, but I've been advocating that we should start vaccinating immunocompromised patients now. I think they should get a booster dose, in my opinion, but that's just my opinion. That's not.
Well, that's what my immunologist is waiting for a little bit more data. He's pretty sure that he's going to give his patients a booster dose as well, or suggest that we get them. So
I think we have enough data now to act on immunocompromised people and give them a booster dose. It's good enough for me.
Okay.
But that doesn't mean it's good enough for the FDA and the CDC, and they're looking at a much broader amount of. This is such a safe vaccine, and I think the benefits far outweigh the risks. We already know we can give it in immunocompromised people, so I don't really see the hesitancy to give a booster. The reason that we do this all the time with other vaccines, right? And we have more evidence for this, right, than other ones. So
yes.
I hopefully you'll get a booster. I would recommend that when it becomes available, get a booster.
Okay, thank you. And is it possible to get a copy of your presentation? Because I would like to read everything. I don't know, Ashley, if we can do that.
Ashley has a copy, so she could send it to you.
Okay, thank you.
If she doesn't mind. She has your emails.
I will do that.
Thank you.